Hello, welcome to Sense of School of Life Sciences. Today we shall talk about histone modification and epigenetics. What is epigenetics? Epigenetics is a study of heritable changes in gene expression that do not involve changes in DNA sequence. Generally we know gene expression will be changed only by any change in DNA sequence by mutation, etc. Isn't it? While epigenetics deals with the heritable changes in gene expression that do not involve change in DNA sequence. That is, epigenetics means a change in phenotype without a change in genotype. Histone modification play an important role in epigenetic regulation. Epigenetic change can also be influenced by several factors, including age, the environmental, lifestyle, DNA methylation, X chromosome inactivation, etc. That we will study in separate class, especially DNA methylation, deacetylation, phosphorylation, etc. We will study in role of chromatin in gene expression in separate class. So here, histone modification is one of the main reasons for epigenetics. Okay, histone code hypothesis states that gene regulation is partly dependent on histone modifications that primarily occur on histone tail that I will say later. Transcription of DNA is largely regulated by post-translational modification to these histone. Before we study the histone modification, we can study the structure of histone. Eukaryotic chromosome structure and its organization is much more intricate, it's much more complex. There is a set of positively charged basic proteins called histones. I will show the diagram, then that will be more clear. You see? There's a set of positively charged basic proteins called histones. Histones are positively charged and basic. Why? Because histones are rich in the basic positively charged amino acids, namely lysine and arginine. Lysine and arginines are rich. The histones are organized to form a unit of 8 molecules called histone. See, the H2A, H2B, H3, H4, 2 each. So 4 different histone. Here 2 each. Thus histone octamer is there, and it is wrapped around it by DNA. So this is a nucleosome. So what is a nucleosome? A histone octamer wrapped by DNA. Okay. Then histones are organized to form a unit of 8 molecules as histone called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called a nucleosome. Nucleosome, it is a 10 nanometer structure. So it's a 10 nanometer fiber. So here negative and positive charge will attract. Just remember like that. The octamer of protein consists of 2 molecules, each of the 4 different histones. Like, what are the different 4 histones? H2A, H2B, H3, H4, 2 each. The core particles, that is this histone octamer. These histones, they are linked by DNA, which in turn is associated with only one type of histone. Here, this is the DNA which is wrapped around it. It is the linker DNA which link the adjacent nucleosomes. So what is a nucleosome? A histone octamer wrapped by DNA molecule. This is a histone. So likewise, numerous histones are there. This is 1, 2. I will show this. You see, 1, 2, 3, 4, 5, 6, 7. Like this, numerous histones are there. Understand? These histones are linked by linker DNA. You see, 2 adjacent histones are linked by linker DNA. This is linker DNA. Okay. So in the linker DNA, there is a special type of histone, maybe H1. So that will be more clear here. You see, this, I told you, these are all series of nucleosomes linked together by linker DNA. And this appears to be what beads on a string structure. Is it beads on a string structure like this, isn't it? So this much part of this beads on structure of the nucleosome is enlarged here. You see, this much region is enlarged here. This much region. Clear? Here, three adjacent nucleosomes are here. So this histone octamer wrapped by DNA. This is another nucleosome wrapped by DNA. It's also here. Then adjacent nucleosomes are linked by linker DNA. You see, linker DNA. Here it is the linker DNA. In the linker DNA, there is H1. See, there is H1. So H1 is seen in the linker DNA. Clear? Clear? Okay. Another very important thing. This I told you, this 10 nanometer fiber. What nucleosome? Nucleosome, it is 10 nanometer fiber. But this nucleosome undergo further condensation to form 30 nanometer structure. That is solenoid structure. What you call solenoid structure? The nucleosomes undergo further condensation to form solenoid 30 nanometer structure. The solenoid structure undergoes further coiling to form the chromatin fiber of chromosome. So a chromosome contains the chromatin fiber, which in turn was formed from solenoid structure. Okay. So nucleosome with a 10 nanometer structure is more open and accessible and biologically active. Solenoid, which is 30 nanometer structure, is repressive structure. I think it is more clear. Okay. Then we can study histone modification. First of all, acetylation. See, histone acetylation is one type of chromatin modification, which may lead to alteration of the chromatin, which eventually change the gene expression pattern. In histone modification, the lysine residue with the N-terminal tail, I will show now, N-terminal tail protruding from the histone are acetylated. The enzyme for acetylation is histone acetyl transferase. I will show the diagram. See, this is a nucleosome. The histone tail you should understand. That's why I have shown this diagram. Clearly understand. See, histone octamer S3, S2A, S2B, S4, and the histone octamer. These histones have a tail. You know, the protein, after all, it's a protein. You know, a protein has N-terminus and a C-terminus, isn't it? It is the N-terminus tail of H4. Likewise, all these histones are has tail. Histone tail, this is N-terminal. Understand? And it is the DNA, it is seen around it, wrapped around it. The figure, it is clear, isn't it? So, in histone acetylation, the lysine residue within the N-terminal tail protruding from the histone are acetylated. You know, here K5, K8, K12, K16. You can see now is a K20, like that. It is numbered. What it is? The position of this lysine. Lysine, K, one letter symbol for lysine is K. Everybody may know. Arginine, it is R. Arginine, it is R, like that. So all these are important when we study biochemistry. This one letter symbol, etc. I think you know this. Okay. So in acetylation, lysine residue within the N-terminus tail. You understood this N-terminal tail. Lysine residue within the N-terminal tail protruding from the histone are acetylated. The enzyme is what? Is enzyme for acetylation? Histone acetyl transferase, or HAT. Very important. The enzymes are very important. when an acetyl group is added to lysine, its a positive charge is neutralized. Thus, the overall affinity of histone for negatively charged DNA is reduced. In this configuration, the DNA is more accessible for transcription factors. Thus, a chromatin remodeling will occur there, and the transcription is increased. Acetylated histone lead to relaxation of DNA and are associated with the euchromatin. You know the euchromatin and heterochromatin. Euchromatin is genetically active than heterochromatin. You know. So once more, when the DNA, histone get acetylated, the overall affinity of histone for negatively charged DNA is reduced. In this configuration, what happens? DNA is more accessible for transcription factors and transcription is increased. It is acetylation. Okay. Then acetylated histone in specific residue creates histone code. Hear me? Acetylated histone in specific residue creates histone code. Different you see, lysine residues are here. K5 is lysine, K represented lysine, K5, K8, that is lysine in the 8th position, lysine in the 12th position, lysine in the 16th position, like that. You see, acetylated histone in specific residue creates a histone code. Then this histone code can be recognized by an acetylated it can be recognized by it's a bromo domain, very, very important. So histone code can be read by bromo domain containing protein. That is, it can recognize the acetylated lysine residue. Okay, then where does the particular histone acetylation take place has different meaning. You hear me? It is the 16th position. Suppose if it is acetylated, it has one meaning. Epigenetic modification is in one way, and epigenetic modification will be in another way if it is acetylated, the 5th position of the lysine. 5th position, lysine and histone. Okay, with an example that will be very clear. You see, it's an example. H4K16AC. What do you mean by that? H4K16AC. H4, that is in N-terminal tail of H4 lysine. N-terminal tail of H4 lysine. This is H4 lysine. Lysine in the 16th position, H4, like in the N-terminal tail of H4 histone, in the N-terminal tail of H4 histone, lysine in the 16th position, get acetylated. That's the meaning. You see, H4K16AC. If it is methylated, then Me, we will say Me. Okay. So methylation is not in the lysine 16th position. So code that is like this. See, when H4 lysine 16 is acetylated, the chromatin tends to form the less condensed bead zone string configuration. Bead zone string configuration is less condensed. You know, it is on condensation we get the solenoid more compact structure, and that in turn compaction will get for the chromatin fiber. Okay, so it is less condensed. So if acetylation take place lysine in another position, I told you that the epigenetic modification may be of different types. Clearly understood, I think. See, when histones are deacetylated, removal of this acetyl group, the positive charges are restored and compaction of chromatin occur. Okay, deacetylation. Deacetylation leads to a repression of transcription because the necessary transcription factors, regulatory factors, and RNA polymerase complex are unable to gain access to the DNA. The enzyme for deacetylation is histone deacetylase, HDAC. That also is very important. Enzymes are very important. So this is opposite of acetylation. Now we can see another modification, that is methylation. See, histones can be methylated on lysine and arginine. Arginine, one letter code, I told you R. And acetylation is only on lysine. Remember, it's very important. Histones are as methylated on lysine and arginine residues only. Methylation is most commonly observed on lysine residues of which in H3 and H4 histone tails. Okay, lysine can be mono, di, or trimethylated. You see, it is mono one, monomethylated or dimethylated position. It is trimethylated. The enzyme for methylation of lysine is histone methyl transferase. Histone methyl transferase. Okay. Then, unlike acetylation, methylation does not change its positive charge. Okay. Generally, methylation of an arginine residue requires the enzyme protein arginine methyl transfer, PRMT. It is similar to HMT, and I will say only the protein arginine methyl transfer and usually PRMT. Methylation and acetylation basically are working opposite to each other. Remember, methylation leads to compaction of chromatin and demethylation would lead to a relaxation of chromatin. What about the acetylation? While acetylation, in acetylation, relaxation of chromatin occurs, and deacetylation causes compaction, isn't it? So that is methylation and acetylation basically are working opposite to each other. Very important. Then about phosphorylation, we can say one word is very important. Phosphorylation of histone in detail I am not going to discuss. It's an important, very important point. Phosphorylation of histone. Unlike acetylation, phosphorylation targets serine, threonine, tyrosine, etc. That is also very, very important. It is also asked in several exams. So phosphorylation of histone. Unlike acetylation, phosphorylation targets where serine, threonine, and tyrosine. Okay. The DNA methylation. About the DNA methylation, actually, we will study this DNA methylation in role of chromatin in gene expression. But here, DNA methylation has a correlation with the deacetylation of histone. That is why I mentioned DNA methylation here. DNA methylation occurs at CPG sites, that is cytosine phosphate guanine. CPG sites are commonly referred to as CPG islands. So CPG islands are regions with a high concentration of cytosine guanine pairs linked by phosphate. So cytosine in the sequence CG of DNA gets methylated to form 5-methyl cytosine because methylation occurs at the position 5-methyl cytosine. Remember, it is interesting to note that methylation of DNA correlates with the deacetylation of histone. Remember, but methylation of DNA correlates with the deacetylation of histone. This provides a double means for repression of genes. Another very important aspect about it that also I mentioned here, methylated cytosine is sometimes known as fifth base of DNA. What we call the fifth base of DNA is methylated cytosine. It is attracting increasing attention as a potential biomarker. Just a familiar example, epigenetic age is higher among people with the obesity. That point also you just remember. Then histone code, what it is. What is histone code? We studied several aspects about the histone code. In short, histone code is written by histone acetyl transferase, isn't it? It's by acetylation, relaxation of chromatin occur, and chromatin remodeling occur, isn't it? So histone code is written by histone acetyltransferase. This acetylated histone can be recognized and bind with the bromodomain containing protein. So this bromodomain containing protein, it can recognize this acetylated histone. Thus histone code can be read. Then histone code can be erased by histone deacetylase, isn't it? So it is in short about histone code. So we can do some questions. See, it's a simple question. It is asked in CSR net exam. Histones are rich in amino acids. It's a very simple question. Arginine and lysine. So another histone deacetylase catalyzes the removal of acetyl group from N-terminal of histone. Which amino acid of histone is involved in this process? It was asked in 2016 June CSR net. That histone deacetylase catalyzes the removal of acetyl group from N-terminal of histone. Which amino acid of histone is involved in this? So this is also a very simple question. It's a lysine, isn't it? It's a direct question. Bromodomain interacts with the RNA molecules, phosphorylated histone proteins, acetylated lysine residues. Acetylated lysine residues are recognized by bromodomain, isn't it? So, one more question. Several proteins are modified by phosphorylation at a specific amino acid residues to alter their activities. Which one of the following amino acids is not typically a site of phosphorylation in protein? Not a typical site of phosphorylation in protein. What are, the phosphorylation targets serine, threonine, tyrosine, etc., isn't it? So here, not typically a site of phosphorylation, it is lysine. Okay. If you like this video, please like, subscribe, share, comment. Thank you.